What's up, everybody? Welcome back to another histology video. It's gonna get a little silly, but if it makes it interesting, that's okay. That's okay. All right, so this video is going to be talking about the histology for Lab 19, the respiratory system. Just want to talk a little bit about the other parts of this lab. So in the beginning, what I would like you to do is read your lab manual from page 169 to 173, talking about the different anatomical structures that you can find in gross anatomy of the respiratory system, so stuff you could probably see with your eyes if your eyes were good, and also identifying that stuff. So from your lab book, there are a number of different models that it's going to ask you to identify. I have copied and pasted them all here, but you can also just use your lab manual if you would like. And I would like you to identify these models. And you're best going to be able to do that. That was a little bit of water. I'm fine. Don't worry about me. You're best going to be able to do that by reading the lab manual, taking a look at those identified models, and then watching these 3 videos here, which will go through the gross anatomy of the respiratory system, as well as a closer look at the larynx, which is kind of the more complicated but interesting part of this upper respiratory system. Once you're through with that, I'd like you to go watch this video of a human lung dissection. And I do want to note the video is about a half-hour long, but I want you to skip around in it because a lot of it, this video is made for med students, so they're going to get way more in-depth than I would ever ask you about. But you are going to be able to see a human lung dissection and kind of go through it in the same way that we would have in lab. So in the beginning, make sure you skip ahead to the 3:55-minute mark, and then once you're done answering these questions, skip ahead to the 11-minute mark, answer these questions, identify this picture that I screen grabbed like a pro from the video, answer this last guy, then skip ahead to 19:30 while you answer some more questions, and again, identify a pro shot from that particular video. All right. Histology section below, we are going to have some histological cuts that I grabbed off the intranets. I would like you to identify them, especially these things that are above them. So identify them out of these pictures, and we'll be doing that in just a second on the as the video progresses. But what I'm real interested in after you identify them is this kind of description of them. So this is a little bit coming from the last thing that we did in the lymphatic system, where we had so much histology and it all just kind of looks like grainy pictures of cells. What is going to help you in particular in knowing that when you look at a picture like this is the trachea? So is there things that you're going to find that clue you in? Does something stand out? Does it look like something? There's no wrong answers here. I'm just interested in how you personally are going to remember this histology. So let us begin then with the trachea here. I'm going to make this a little bit bigger so that we can see. For the trachea, I would like you to identify the pseudostratified columnar epithelium, the goblet cells, submucosal glands, hyaline cartilage, and blood vessels. So let's kick back over to this bigger picture of what we can see there. This is a cut of the trachea. Over here, this would be the lumen out here. So this is where the actual air will be flowing. What you see right here is bullcrap. So this is our official name for it is artifacts. So stuff that kind of just gets there in those what would be open tubes when we cut open a person. So this is nothing. Don't worry about this. This wouldn't be here in a living individual. This would all be open air flowing through the middle of the trachea, aka the windpipe. So thus, our epithelium, which would be the cells directly interacting with that external environment, would be right here. So here's the very bottom of our fake layer. Remember, pseudo means fake. So our pseudo-stratified columnar epithelium. And you can see the nuclei of them right there. So nuclei, nuclei, nuclei. Remember, each one of these cells will go down and touch the basement membrane down here. What you can also see are these clear areas here. These actually aren't artifacts. These would be where the goblet cells are kind of stuck in between our pseudostratified columnar epithelium. So there are the goblet cells, the kind of clear area right there. Going down below this layer, we get to all this connective tissue down here. This is called the submucosa. There's a lot going on in this area. We definitely are going to find some blood vessels. There's one right there. We can tell because we've got some pale red blood cells in there, and it's surrounded by a simple squamous epithelium. So there's a blood vessel. There's a blood vessel. We got some major blood vessels in this area. Another thing that we have to find, let me get this work, there we go. So we found our pseudostratified columnar epithelium, we found our goblet cells, we found our blood vessels. We also want to find submucosal glands and hyaline cartilage. How do we find glands? Well, we remember how to find glands. Glands are made of a simple cuboidal epithelium. So we got to look for cube-like cells, and bada bing, bada boom, we've got us a bunch of cube-like cells right here. So this submucosal gland here would be creating liquid that would run through a duct that would curl its way all the way out. Oh, gosh. Sorry, that's my dog, Luna. And she barks at this time every day when the milk arrives. What is this, 1930s? She barks like this when the mail arrives. Give me 2 shakes and I will stop her barking and come back to the histology. Okay, my heart rate is down again. My dog is silenced. Let's continue. So we were just talking about these simple cuboidal clumps over here. This is our submucosal gland. The last thing to find is hyaline cartilage. And we know, because we are awesome anatomy students, that we can find cartilage easy because those cells have lacuna. It's got that little open space around them. And bada bing, bada boom, what do we find down here? Cells with lacuna. It's also very glassy in appearance, very smooth, and that is hyaline cartilage. So you got your hyaline cartilage down here. That's what you can feel if you find your Adam's apple right in the middle of your neck and you go down below it and you press in and you feel something hard and you're kind of choking yourself and it hurts. That is this cartilage that you're feeling right there, keeping your airway open so that air can flow to your lungs and you can make awesome videos like this. Let's continue on. The next thing we're going to look at, remember to give me a description of how you'll remember. If I show you a picture like this, I won't show you this picture. That'd be way too easy. But if I show you a picture like this, how would you know you're looking at the trachea? How would you find the things in it? As you move from the trachea, you then get into the big tubes that are taking air to the sections of the lung. Those are the bronchus, or the bronchi for plural. So here is the bronchi. What we're going to find are cartilage plates, smooth muscle, our pseudostratified columnar epithelium, and some goblet cells. Let's get to it. All right. So here we have our bronchus. And let me tell you, this is why this slide in particular is awesome. This is why it's hot. This is why it's hot. This is why it's hot. If you watch my lecture video on the respiratory system, you will note that by the time you get to the bronchi, so the primary and the secondary, especially in the secondary, the cartilage that surrounds these tubes is not going to be a complete circle like cartilage. It's not going to be C-shaped like it was in the trachea. It's going to be irregular plates that kind of surround the outside. And if you look at this picture, that is what is so hot about it. Around the bronchus here, we don't have a complete ring or a C. We've got these boop, 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 kind of irregularly spaced cartilages. These are the irregular plates of cartilage that surround the bronchus. That's why it's so cool. Dude, you have no idea. I have yet to have found a picture that shows this awesome kind of irregularly spaced plates of cartilage, and here it is. So there it is. What else we got to find? We got to find smooth muscle. How do we find smooth muscle? We think back. We think back to our previous histology. It's going to be red in color from the staining. It's going to be stretched out. The nuclei are going to look real stretched. It's going to look like a football that has been deflated. Look out, Tom Brady. I went there. Deflategate. So, where do we find that tissue? Right here. So here is the smooth muscle that goes around the bronchus. Very important because it changes the size of the bronchus. It either allows more or less air to go through this to get to the lungs, depending on the situation. So there's that smooth muscle surrounding the outside of the bronchus. As we move interior, we once again find ourselves the pseudostratified columnar epithelium. And anywhere that we look and we find these kind of clear goblet-like patches, so goblet-like patches, those are going to be our goblet cells. Boom! Bronchus. So the bronchus, cartilage plates, smooth muscle, pseudostratified columnar epithelia, and the goblet cells. Let's continue down this tube. So we were going down the trachea, now we're going down a bronchus, now we're going even further till we're getting to the very tiny microscopic type of anatomy that is diverting air and also going to be exchanging air. Finally, so we're finally going to get to a place where we're going to be exchanging carbon dioxide and oxygen. So this picture, this final picture, is going to be a lot more zoomed in than our previous picture was. All right, so this is zoomed out. This here is going to be something like a scanning microscope or something like that. This is you're getting into your hot power 400 times magnification, even more stuff like that. So we are zoomed in here, zoomed out here. So we have zoomed into smaller structures. In this case, we're going to be looking at the bronchioles and the alveolus. So we're going to find smooth muscle, pseudostratified columnar epithelium, and goblet cells on the bronchiole. And then we're going to find an alveolus, type 1 alveolar cells, and even a blood vessel, the very thing that we are going to be exchanging gas with. So bam, let's come back here. If you remember our bronchus, and who could forget it, the bronchus is very circular in structure. It's got the pseudostratified columnar epithelia, it's got smooth muscle, it's got plates of cartilage. Look in here, there's one structure that mimics that very well, and that's going to be this bad boy right here, a bronchiole. So a bronchiole is still going to have our pseudostratified columnar epithelium. You can see the little wispy hairs there a little bit. It's getting a little bit harder to see, but they're still there. You can definitely tell that the nuclei are at all kinds of different levels. So this is going to be that pseudostratified columnar epithelium. We're not really going to see as many goblet cells here, so I didn't put that into the requirement. requests, but we do still see around the outside that smooth muscle. So the smooth muscle has continued around these smaller tubes to open or close them, depending on if you need air or if you don't need air. What has disappeared, though, very important, what's disappeared? Cartilage. So by the time you get down to these microscopic bronchioles, you no longer have that cartilage anymore. Looking all around the microscopic bronchioles here, we're going to find many alveolar sacs, type 1 alveolar cells, and even some blood vessels. So looking here, we need to find ourselves, oh, I had it there. No way. We're going to get rid of it. It's too hard to find. All right, no goblet cells. All right, coming out here, we're going to be looking at an alveolus. So here's just a bit of, let's see, a bit of vocabulary. The bronchioles are going to get smaller and smaller and smaller until they become respiratory bronchioles, which is what you see right here. And a respiratory bronchiole will lead into one of these big kind of grape-like clusters here. And this grape-like cluster is an alveolar sac. Alveolar. Uh-oh. Uh-oh. It's harder than it looks. Give it a try. Alveolar sac. Boop. All right, the alveolar sac is made up of individual alveoluses. Alveoli for plural, alveolus for single. So this right here, boop, this is an alveolus, which is what you see right here. So just one of those little dimples in this alveolar sac is an alveolus. Here's a nice one right here, another one right here. So these are all alveolus. Believe it or not, an alveolus is made of type 1 alveolar cells. Mind blown. So each of these nuclei that you see here are the nuclei of a simple squamous cell that at this point is called a type 1 alveolar cell. They are simple squamous cells, so they're not any different from other simple squamous cells that you find elsewhere. It's just because they're found in the lung, they're here, we give them a special name. These are all type 1 alveolar cells. The last thing that we have to find is a blood vessel. Blood vessel is also going to be surrounded by simple squamous cells. Here's a simple squamous cell. Here's a simple squamous cell. The only difference between an alveolus surrounded by simple squamous cells and a blood vessel surrounded by simple squamous cells is the alveolus is going to be open because that's where air is, and the blood vessel is going to be containing blood. Believe it or not, the blood vessel has red blood cells in it. So here's some blood vessels with red blood cells inside them, right next to an open, open alveolus. And so we are going to be getting exchange of obviously green oxygen into the blood. And as we all know, carbon dioxide is going to be, we all know that's orange, right? So CO2 will be leaving to come out with our breath out. So there you go. There are the individual cells, structures of the histology of the respiratory system. Have a good day. Be good to each other. Contact me if you have any questions. Hope you had a larf, and I'll see you later. Adios.